and welcome to the Research Goes Viral by the MRC, University of Glasgow Center for Virus Research podcast. My name is Bailey Atkinson. I am a second-year PhD student based with Liam Brierley and Dr. Daniel Stryker's groups. And we're here today with the 12th Sir Michael Stoker CVR Award of 2025 recipient, Professor Akiko Iwasaki. Welcome.
Thank you so much. Yeah, great to be here.
Absolutely. And Charlotte, I'm wondering if you could tell our listeners what this Sir Michael Award entails.
So, yeah, absolutely. Essentially, it's an award given to people who have made significant contributions in the field of virology or immunology. And the special thing about it is that the award is voted for by early career researchers only. So PhD students, postdocs, and technicians, essentially. And the recipient of the award essentially gets to visit the CVR and really have direct interactions with early career researchers in a way that emphasizes, those connections. And it's a really, fantastic scheme, and you are a very, very worthy winner. So we're really glad to have you here.
Thank you so much. What an honor.
So, yeah. And so before we get to the main part of the podcast, we usually like to start out with like a very brief academic introduction. Mm-hmm. So I'm going to go a little bit of a biography of your history, and feel free to correct me if anything I say is incorrect, or if you want to add any other information as I go along as well. So Akiko, you got your Bachelor's of Science in Biochemistry from the University of Toronto, and then your PhD as well from the University of Toronto in Immunology. You then went and worked at the NIH as a postdoc for 2 years, and then moved to where you are now at the Yale University School of Medicine, working as a Sterling Professor of Immunobiology. You also noticed that you were an investigator at the Howard Hughes Medical Institute, which I haven't heard of before, admittedly, but I looked it up. So for those that also haven't heard it before, it's a biomedical research organization and a philanthropy organization as well.
Yes. Yeah, it's such a privilege to be an investigator at the Howard Hughes Medical Institute. They are very generous with funding our research and funding my salary and just, providing the opportunity to interact with other investigators.
Yeah, that's fantastic. So my first question that I had for you is something that I've also been experiencing actually in my own lab, where when talking to researchers here or just in life, a lot of times you hear about how their first research focus when they start a group, or even if they're in a group for a long time, it ends up shifting their focus over the years. So for example, in my group, we focus a lot on vampire bat rabies, but now we've been focusing more on other viruses that are exposed, or rather transmitted from bats. And then looking at other virus reservoirs like rodents, for example. We've even looked at avian influenza. So the change has been quite noticeable. And I noticed this is something that you've had to experience for yourself, for your group, specifically due to the COVID pandemic. So I noticed before the pandemic, your lab focused more on the mechanisms of immune defenses against viruses at mucosal surfaces like respiratory or reproductive tracts. So when COVID-19 hit, I read that your lab had to make that research pivot in the matter of weeks, like very fast. So I'm wondering if you could walk me through what that process was like for you, and how did you decide which questions on COVID to pursue first?
Oh, yeah. So these are all great questions. I have, throughout the 25 years of having my lab, shifted my question over, I would say about every 5 years or so. There are new questions that come up, either because there's a new virus that emerges, or new questions that somehow pop in my head, or whatever it might be. So it is true that the shifting of the focus is also fun, because you get to think about a whole new set of ideas and questions. But for COVID, it was more dramatic and drastic than my usual shifts, which really happened overnight, when the pandemic was declared by the WHO, and then soon after we had to start dealing with lockdowns and the virus coming from China to everywhere and how quickly it was spreading. And so, fortunately, because we have a lot of collaboration within the Yale University School of Medicine with investigators in medicine and investigators in the public health department, we were able to form a bioethics approval for conducting human research. As soon as the first patient was admitted to the hospital, we were there. So we were from N of 1, like the first patient, all the way to, we even have a protocol right now to collect patient samples. So it's been great to gather our sort of efforts together to get this institutional requirements that were already set up to start analyzing the first patients. So that was great, and that's when we started to collect saliva samples, blood samples, fecal and urine samples to be able to investigate what's happening in patients. Yeah. And we also had lots of lab members came to the lab, and because it was locked down for any other research, people who were working on unrelated viruses like herpes came to the lab and contributed to the surveillance PCR testing that we were doing for the school, day in and day out. It was like a very well organized operation that we did for the school, and that was also meaningful for the people who were doing it. But yeah, so that's how quickly we had to shift. It's basically an overnight.
Wow, it's really exciting, but also I would assume very stressful to experience that.
Yeah, definitely. Yeah, all of a sudden, we were on Zoom 24/7. Yeah. But yes, it was stressful, but meaningful and rewarding.
Yeah. Definitely. No, that's really kind of interesting to hear about. It was a similar, situation here where it was very much, as you said, an overnight pivot, and came with some quite challenging aspects, I can imagine. But that's fantastic. So in terms of kind of the research that you've done, it's been absolutely fascinating just to, read about and to see it developing. And kind of a big part of your kind of research, of course, is understanding kind of the pathogenesis and the actual root cause of long COVID. So what are your current hypotheses as to what kind of causes this range of symptoms to occur in patients?
Yeah. So this is the question I think about, day and night. What are the root causes of long COVID? Because by answering that question, we will be able to treat patients with appropriate medicines. Okay. So there are, currently four major root cause hypotheses. And downstream of that, there are other things that can happen, like, clotting issues or other sort of neurological issues. But root causes, what we're thinking about is if you treat them, you should be able to cure them, right? So those are the causes we're interested in, and one of them is persistent virus. So there's evidence now from 100 papers that show persistent antigen RNA or combination in tissues, samples, fecal samples, blood samples. You could find evidence of elements of the virus in people with long COVID. But also in people who recover from COVID, there's some evidence of persistent virus in those people too. So whether or not the virus itself that persists causes the long COVID, or it's that plus something else, we don't know yet. But if we can get rid of the persistent virus from people, we might be able to then either cure these people or treat them with additional medicines that may be happening, the additional pathology that may be happening at the same time. Second hypothesis is autoimmunity. So it's very common for acute infection to trigger autoreactive B cells and T cells. And once that happens, it's very difficult to restore homeostatic function in these cells, because once activated, there's really nothing stopping them from being activated. So we see evidence of autoantibodies in a subset of patients. And we even know that they're pathological because we can transfer those antibodies into mice and cause the same symptoms. So yeah, that's when we knew that this wasn't just a biomarker, that cause of pathology. So for those patients, therapies that would reduce the autoantibody, including B-cell depletion therapies or FcRn inhibitors that reduce the circulating autoantibodies, these kinds of medications would be beneficial. And third possibility is that we and others are finding that persistent virus, like herpes viruses, can also reactivate in a subset of patients, and whether that may also be triggering pathology, because those reactivated viruses can replicate infected other cells and cause sort of the symptoms of a viral infection. And to get at that, for instance, we can try to give patients antivirals against Epstein-Barr virus and other herpes viruses. So these are, the third root cause. And the fourth root cause is something that we and others have seen, where people have sort of unrepaired damage from the acute phase of the infection that persists. And that could be either encoded in long-lived cells, like innate immune cells, macrophages, microglia. And when that happens, again, there seems to be this persistent triggering of the inflammation that happens. And those people might benefit from inhibitors of cytokine signaling, like JAK inhibitors. And we have a large clinical trial ongoing right now with people from the Vanderbilt University. So, there are lots of hypotheses and trials out there, and I hope that these will give us more answer to go forward with clinical trials.
Absolutely. Yeah. So many potential avenues, but I mean, I don't know. I'm kind of convinced by all of them.
Yeah, exactly. So the key is that they're probably all true, but not happening in everybody, right? So to identify patients who benefit from drug A versus drug B based on what the drivers are, I think that's where the key barrier right now is in identifying the patients for appropriate drug. But I think there is also some development happening.
Absolutely. Yeah, that's great. When you mentioned I believe it was the second hypothesis that was talking about the reactivation of latent viruses, or is that the third? The third one, yeah. For listeners who might not know, I feel like before I started studying virology, I didn't really know this, that we have dormant viruses that we carry in our bodies. What do you think causes these dormant viruses, to reactivate? And how could that lead to long COVID symptoms?
Yeah. So the viruses that we're interested in, the dormant ones, are the members of the herpes virus family, such as Epstein-Barr virus, as well as varicella zoster virus, which is the chickenpox or the shingles virus, and also herpes simplex viruses, type 1 and type 2, which that's how I started my lab 25 years ago, is studying those viruses. But it comes in handy, that the knowledge that we accumulated over the years, because now we are suddenly, facing long COVID and potential involvement of herpes viruses. So in healthy individuals, so we acquire these viruses fairly early in life. And when an infant or toddler gets these viruses, it just looks like the flu or the common cold, or, the symptoms are not obvious to people, parents and physicians, and usually isn't even diagnosed. But what happens is these viruses can become latent in cells, such as neurons and B cells. And when that happens, it's impossible to get rid of them because to the immune system, it's invisible. Just the genome is saying that quietly. There's nothing, no handle. They can even latch onto. But these latent dormant viruses become reactivated in response to certain stress, such as sunlight or even physical, emotional stress can cause this. And sometimes the immunodeficiency can reactivate these viruses. And that's maybe what's happening in COVID is that some people, reactivate these dormant viruses in response to this acute infection with SARS-CoV-2, which is, quite stressful for the immune system. Yep. And we know that lymphopenia or the reduction in the number of lymphocytes happen in especially in severe cases. And when that happens, what's keeping those viruses dormant also are lifted, and these viruses reactivate, and they can then replicate and infect other cells. And so, most people who are not interested in virology may not know that we're all living with these viruses in latent forms. But, for example, herpes simplex type 1 can manifest as cold sores. For those of you who experience cold sores, they often happen when there is a stress.
Right.
And so the immune system is no longer able to keep those viruses down, and they come out. So this may be sort of what's happening in a subset of people with long COVID, is that these reactivated viruses are now then becoming pathological.
Yeah, kind of tying back to our first topic of the conversation, it was cool to see that the work that you were initially doing with herpes viruses and stuff is now kind of coming back into your work with long COVID. It's all full circle.
It totally is. And you never know, right?
Yeah.
You never know what you're studying now could be useful 20 years down the road. And it's really rewarding to be in a space where you can utilize your previous knowledge to benefit people today.
Yeah, definitely. No, the interplay between kind of these viruses is really fascinating and definitely kind of an underexplored field. So it's kind of really, really interesting that you kind of feel that that's one of your hypotheses, because it's, very, very interesting. So in terms of kind of SARS-CoV-2 itself, you found evidence of kind of viral reservoirs in the body, or kind of pieces of the virus persisting in the body. If I recall correctly, you found it kind of in the intestines, for example, of patients suffering from long COVID, which is, really interesting. So how did you actually discover this? How did you come about detecting this? And when we kind of think of the treatment that we could offer to kind of long COVID patients, what kind of implications could that have?
Yeah, so it's not me that discovered this, but the field itself. There are lots of papers now showing biopsies and autopsy of people who died with long COVID. And you can see viral RNA in the intestine, viral RNA in lymph nodes, and many other organs. And also proteins in the skin, the lymph node, the many places, including even the bloodstream. So initially, I think it was Dr. David Walt's group who reported spike protein in the blood of people who had COVID in the past. And that was, surprising because, in the textbook, explanation of coronaviruses is that you get infected, you recover, you get rid of the virus altogether, and you move on. But that's not what's happening here. Is that some people continue to carry the virus, or at least fragments of the virus, so that they are producing the protein. And in later study with Dr. Michael Caruso, they demonstrated that viral spike protein can be detected in 30 to 40% of people with long COVID, and about 15% or so for people who recovered from COVID. And so, even though it's not a black and white sort of discrimination factor for long COVID, the fact that long COVID patients have higher levels, higher frequencies, suggests that the spike protein or anything related to the virus, persistent virus, may be driving this disease. So this is a very important finding, because what we can do with that finding is that we may be able to identify people who have persistent virus for a clinical trial against, let's say, using antiviral drugs, or using monoclonal antibodies that target the virus. And whereas people without this signal may not benefit from such treatment, right? And they may instead benefit from B-cell depletion therapies against autoantibodies and things. So this kind of marker that directly reflects the root causes is really, really important.
Absolutely. Yeah. That's so interesting. And I think kind of going into thinking about the future for the most promising research direction to take with long COVID, would you say it would be developing these different kinds of medications for these patients that are experiencing these potentially different kind of outcomes with long COVID?
Yeah.
Or what do you think would be more promising research direction?
Oh, there's so much to do.
Yeah.
So much to do. But as you say, Billy, it's very important for the field now to be testing different drugs that get at these root causes using randomized clinical trials. The most rigorous form of testing a drug in people as soon as possible. Because, as I mentioned, everyone may have different drivers going on. For example, if a person A has persistent virus and person B has autoantibody, person C has both. If we can identify their biomarkers and identify the root causes, then we can give patient A the antiviral drugs, right? Patient B the B-cell depletion therapies, and patient C might need both.
Yeah.
Yeah. So this is the kind of strategy that we need to take even like a platform, randomized clinical approaches to be able to test drugs one after the other in patients in sequence until we find the right drug for the patients. Or we can do individual randomized clinical trials and then try to find out biomarkers of responsiveness. And because, for instance, we were involved in a Paxlovid trial at Yale. And this was a great trial because we did it in a decentralized manner, which means that we can recruit patients electronically. We didn't have to have them come into any clinic. We sent the phlebotomist to their homes, collected their blood. Yeah, we sent the drugs to their homes. And so they didn't have to go outside of their homes to be able to enroll.
It's fantastic.
This is really important.
Yeah, super accessible.
But yeah, many patients are too ill to travel. Yeah. And those are the patients we need to help, right? So this was a great trial. And even though the outcome of the trial was negative, meaning 15-day course of Paxlovid was not sufficient to improve the health of these people, we learned so much doing biological analysis before, during, and after to learn who benefited from these medications and why.
Yeah.
And that's what we need to be doing, like tens or hundreds of these trials. So we can get to the answer quick.
Yes. Wow. Yeah, that's so important, the accessibility of clinical trials. And, it's fantastic that there was kind of a way to do that. And especially if something as multifactorial as long COVID, it's so important to keep pushing those kind of trials. So it's really fantastic that, there are ways to do that will impact the most affected patients as well. Thank you so much for that. And I suppose that our final question, we would just like to say congratulations on your award of the Sir Michael Stoker Award, and we've been so happy to have you here. It's been fantastic to kind of talk to you and learn more about this. And we were wondering, have you had a chance to explore kind of Glasgow or Scotland itself at all before?
Well, before I visited Edinburgh a long time ago. That was a wonderful experience. And yesterday I had a chance to walk around Glasgow a little bit and went to the Kelvin Grove Museum.
Beautiful.
Beautiful. Yeah. Oh gosh, I needed a whole day there.
Yeah. And it's free.
And it's free. It's wonderful.
Yeah.
And so I got a little taste of Glasgow, which is wonderful. And thank you so much for hosting me. It's such an honor for me to be here.
Absolutely.
Absolutely.
Yeah. And if you're ever interested in looking for Highland cows, if you have,
That's what I want to see.
yes, I recommend going to, it's called Pollock Park in the south of the river. It's fantastic. They're always out. I remember the last time I went, which was maybe about a month ago, they still have some young ones. So, and they're very curious.
Yes, they're very friendly.
Definitely on my bucket list.
Yeah. Absolutely. Yeah.
For that recommendation.
Of course. Absolutely. And thank you everyone for listening to the podcast. Thank you again, Akiko, for being a part of it. And yeah, I hope you have a great rest of your time here in Glasgow.
Thank you so much.